So the next step is to perform the trimming. In general, the data is of good quality as we saw, but perhaps in the end we need to do some trimming. So now we will use Trim-O-Matic. Trim-O-Matic is similar to SQL that we had used last time, and the data that we have is singlet. So we choose here singlet, and I go here to multiple data set, and I choose all of them. So we have 2 input and 2 control, 2 input and 2 input here, and 2 with immunoprecipitate. From the fastqc we saw that probably we don't have adapters, so here I choose no. And now these options are similar to what we had in SQL. So Trim-O-Matic, like SQL, calculates the average quality for so many base pairs as we see here. So here is the number 4, and if the average quality is below this number here, which is 20, then it removes this small window of 4 bases. And so we will just leave the default as they are, and I will just click on Run Tool. Let's wait for a while, and then we will do fastqc again. Okay, trimming is going on. We just wait for one file, but meanwhile I will just do a fastqc for just one of these files. So go again, fastqc. And I have a collection of data. So we'll choose .26 is not finished yet, so maybe I will just not put it here. And everything else is default, so now I will just click on Run Tool. And let's wait to see the quality control. Okay, so we have now some results from trimming. So we see here the trimmed data, the trimmed file. I don't remember which one is that, which one we had seen previously, but the quality now is above 32 for all the length of the read, so we are pretty good. The rest are still running, and we see here that some trimming is taking place because not all reads have exactly the same length. So here is the sequence length distribution, and here is the length, and here is how many reads have the respective length. So for example, if no trimming would have happened, then all reads would have this length here, but since some trimming has happened, some reads are a bit shorter. Okay, the rest are in fact okay. We can see one more, this one. This is even better actually. The quality is pretty high. And again here probably some trimming has taken place, just a little bit, or for very few reads here. Okay, and we have one more, and also here the quality is very good. Okay, now we can proceed with the next step, which is the mapping step. So we have different options there, either to use a tool called the tool that we had used previous time called BWA, or to use the bow tie tool. So if I remember well, in the original paper for this dataset they used bow tie tool, but in the tutorial that we follow, this one, they use BWA. So I guess we will also go with a BWA option. Now if some students want to follow the bow tie 2 version, it will be useful because we will be able to compare the results. Okay, so I go for the BWA. So we choose this tool here, map with BWA. We read from the description that this tool should be used when the reads are less than 100 base pairs. We saw in the report of fastqc that the reads are about 50 base pairs what we have, so we'll go for this one. And let's see what we have here. Will you select a reference genome with history or built-in? Let's use a built-in because these data are from mice, so the reference is pretty good here. Now we can choose MM9. They don't provide the MM10, so we'll go for the no, they provide the MM10. If I remember well, in the paper they had used the MM9, but we can go for the MM10. Okay, the data that we have is single end, so single fastq, and we can go to align all of them. So these are the results of Tremomatic. Let's be careful here. 2 with the antibody and 2 with the control. Okay. Everything else probably will remain as a control. So let's choose this again. Okay, built-in MM10, our 4 files, and everything else is the default, and just click run. Okay, we have to wait. Now, while we are waiting, I can go on and describe the next step, which will be the assessment of chip quality here. So what we have is 2 files. These are biological replicates. With TAL1 antibody, so these are precipitation has happened here, immunoprecipitation has happened here, and 2 files without immunoprecipitation, just the input. Okay, and what we'll check now is how these files are correlated to each other. So let's try to draw that. Okay, so we have, so let's say that this is our TAL1. This will be our TAL2, TAL1 again, 2 file, and then we have the input one. So from the same sample as this one, they just split the input in 2 parts. In one they did the immunoprecipitation, in the other one they did not do the immunoprecipitation. So this one, the first one is a TAL1, and the second without the immunoprecipitation is the input one. The same with TAL1-2, the second sample, the second biological sample. In one they did the immunoprecipitation, this one, and in the other one they proceeded without the immunoprecipitation. Now, when we do the mapping, so if this is some gene here and this is another gene, maybe we'll have something like that. So some reads here, some reads here. So let's assume that transcription factor binds here and this TAL1 binds here. And here we'll just have, could be again genes here, but since we don't have we haven't performed the immunoprecipitation step, we'll just have some reads randomly depending how accessible the chromatin is. Something like that. Now this control step, this chip quality step, what it does is that splits the genome in bins So you can imagine that it splits in pieces, like one piece is this one, the second piece is this one, the third piece is this one, the fourth piece is this one, the sixth piece is this one. So let's just put 6 pieces. And here the same, exactly the same beginnings and ends. Okay, now let's just put some also reads here, just like a noise, and the same is here, true. Everywhere we have some reads. And now what we do is that in every such window here, we just count the number of reads we have, and here it's many. So in the dataset, let's call them tal1, tal2, and here we have input 1 and input 2, and here we have the windows: window 1, window 2, window 3, window 4, and window 5. And if we count now the reads here, we will have, let's say, something like 100 reads, and here we have 125 reads, and here we have 10 reads, and here we have 12 reads, and here we have, let's just put some small numbers here: 5, 4, 4, 1, and let's say 6, 6, 7, 8, and 6, 9, 12, 10, and 6, 6, 5, 4. Because here we have many reads, so that's why I put a big number here. Now what we do next is that we do a correlation between these numbers and these numbers. So this will be a correlation matrix, and here we will have T1, T2, input 1, input 2, and here again T1, T2, input 1, input 2. Now the correlation between these 5 numbers and itself will be 1, and here it will be 1, and here will be 1, and here will be 1. And the correlation between this number and this number, so this vector and this vector, it will be high, because when this is high as well, and when this is low as well. So this number here will be something like, let's say, 0.9, and 0.9 here. And with this way, we will fill all the table. For example, the correlation between here and here will be a bit lower, so let's say that's something like 0.4. And this must be even lower, because this is the same sample, so we expect some correlation. This is without immunoprecipitation, and these are without immunoprecipitation and different samples, so let's say 0.3. Now between these two things, we expect some correlation, because they are both affected mainly by the open chromatin, which must be similar. So here we'll just put the number if I have done it correctly, let's put the number 0.45, and so on and so forth. Okay, now if we want to see that as a program will output that, so this matrix that I created, it's this matrix here, but it is transposed, because here are the samples. Okay, here are different samples, doesn't really matter, it's a different example. And here we have the window, so the window that starts at 0 up to 1000 has so many reads, and the window that starts from 1000 to 2000 has so many reads for the first sample, and so on. And we see that this window has really many reads for the first sample and for this sample here, whereas here and here the reads are a bit smaller. So we'll go on, we'll calculate all the correlations, and we'll do a correlation plot. And okay, we'll see that, but the correlation plot will look like that if we manage to see the figure. Okay, we'll see that in Galaxy immediately. Okay, the mapping has been completed, so let's see if we see some report here. So we have the 4 BAM formats. Let's see if actually we don't see any report here. We'll assess this later on if mapping has been is okay. And let's go for the next tool, that is the correlation plots. Okay, before going to the correlation plot, we can actually inspect the quality of mapping by using a tool called SumTools IDX Stats. So this tool, in order to run this tool, we will do the following. Let me just show that again. So we will use here the 4 BAM files that we have as output from the BWA. So we choose all of them, and then we just click Run Tool. And the report is here. And what we see is a file with 4 columns. The first column is the name of the chromosome. So mice have how many chromosomes? 20 something if I remember well, or yeah, they should be 20 actually, or 19. And then this is the chromosome name. Now just notice here that we have the main chromosomes, but we have also some other pieces. Now this one, for example, it denotes that there is a large piece of DNA, about 170,000 base pairs, that we know that it belongs to chromosome 1, but we don't know where exactly. And that's why they have separated from chromosome 1, and they put it here as a separate chromosome. Let's say as a separate genomic entity. So let's go on for the usual chromosomes. In chromosome 1 has this length in total. So this is about 195 million base pairs, and we have mapped 4,700 reads in chromosome 1. Now this 0 here means that if we could have per dent, this would be the number of the mates that are unmapped. So if, let's say, the forward read would be mapped but the right one not, then this would be 1 here. And if this happens in 100 reads, then this would be 100. Okay, we see that actually we have some mapping, especially in chromosome 19. Seems to be pretty high. Now remember that this is, of course, a downsample file, right? We haven't taken all the whole result of NGS because this would take time. We just sampled it and we took a smaller part of the fastqc file. And that's why these numbers are not very high. But at least we can see that mapping is not very bad. It's okay. And other files have similar output. Okay, since now we are satisfied with the results of mapping, we can go on with the correlation plot. So the correlation will be done with this tool, with a tool called multi-BAM summary. So calculates average read coverages for a list of 2 or more BAM CRAM, it also does the correlation things. The first thing that asks if the sample order matters, no. So let's put all of them. Okay, so yeah, let's do it. Advanced summary. Okay, so here we choose all of them. Now, we explained before that the data set will be split in windows, in what it called bins. So we have 5 bins here, and yeah, it will be in bins. What will be the size of each bin? Let's put 1000 base pairs. And everything else will be default. So we just run it. Here the order of the samples does not matter, so it does not matter which is first and which is last here. And let's wait. So the output of this will go to another tool called plot correlation. Plot correlation to analyze the results. But let's wait until this will finish. Okay, so this has finished. We cannot visualize it because it's a special format that we cannot just view here, but we go to the next tool, which is the plot correlation. Just choose it here. And this is this 44, okay. The correlation, let's do Spearman correlation. We can choose Pearson or Spearman correlation. Let's see what is better. Yeah, let's go for Pearson, doesn't really matter. And the plotting type will be a heat map. And I think everything else is fine, so let's just click on Run Tool. Okay, so now it has finished. If I click on this button, the result of plot correlation, I see this nice heat map. So here I see the let's see if we can make it a bit smaller. No. So this is, unfortunately we cannot see which data is. So this 29, 28, 27, 26. 29 is so this is TAL1, 28 is TAL1, 27 is input, and 26 is input. So these two are immunoprecipitated, while these two are input. And what we see is that the correlation between these two guys is higher. So we see here the correlation between 29 and 29 is 1. The correlation is 1 here. We see the color scale here. The correlation between 29 and 28 is this light blue. Should be something like 0.8, 0.9, so pretty high. The correlation between 29, or let's say 28 and 27 is this light blue, and this is with 26. And you see that the correlation between these two samples is high, and the correlation between these two samples, 27, 26, is high. Which means that when we have high number of reads in this 29 file, which is the first immunoprecipitated data set, then we also have high number of reads in the 28 file, which is the second immunoprecipitated data set. which means that probably things have worked out well. Now this is also correlated between themselves. Why? Because the chromatin probably is more accessible in the same genomic regions. And that's why we see also a quite high correlation between these two guys. Okay, so this is a control that we did just to assess that things have probably worked well. And now we'll do one more control to assess whether we have a good signal in the immunoprecipitated datasets. And with good signal, I mean the following. Let's go back to our notes here. And let's go to the next step. So if we have these files here, let's go to this one. Yeah, if we have the different windows that we had done before, so like these windows here, we can write down that in window 1 we had, for example, in the ChIP dataset, the immunoprecipitated dataset, 3 reads, and in the input file we had also 3 reads. In the second window, so in this window here, in the immunoprecipitated dataset we had 3 reads, and in the input file of the same individual we also had 3 reads. And so on and so forth. We see, for example, here that in the 6th window we had a lot of reads from the immunoprecipitated guy, while in the control, so without the immunoprecipitation, we just had 2. Okay, so we have this list, and what we do now is that we reorder the lines based on this column. So we will put first the row with the smallest number of reads for this ChIP count, which is this 4 row here. This will go first. So this is a 4, 1, 3 row, 4, 1, 3 here. And the second row will be this one, 3, 2, 1, and so on. And what we see is that here in the ChIP count we have small numbers, and suddenly the numbers become big. Why this is important? It is important because in all these windows probably we have an accumulation of reads in a few bins. Let's say like this one here. For the whole genome, this number of bins that have a large number of reads will not be very large. Which means that if we sort them based on the number of reads, we will have a lot of windows with small number of reads in the beginning, and towards the end we will have a few windows with high number of reads, as actually it's seen here. Now what we can do is that we can normalize these numbers. How we can normalize it? If we just we can normalize it by doing the following thing. So the normalization question here is, what is this one as a fraction of all the reads here? So this will be, for example, 1 divided by this sum, and this sum is 113. So this number will be 1 divided by 113, which is this 0.008. Now, in the first 2 bins together, we have 1 plus 2, 3. So the 3 out of 113 will be this number here. And then in the third bin, we have 1 and 2 divided by 113, and it will be this number here. We do this because this number will start from more or less 0 and then will go up to 1, because in the end we will have the sum of all these, which will be 113, divided by 113, which will be 1. And the same thing we do for the input. Now, if you notice these two columns here, you will observe that in the input, the increase in the reads happens gradually, in small steps, just because the reads fall uniformly on the bins. While here, in the beginning is slower, and then suddenly it makes a jump. Now, this is depicted in this way. So this is the control. Okay, this number of reads here is small, so that's why you see this gap, but you know, it goes more or less uniformly along the diagonal, while the immunoprecipitated goes slowly in the beginning and then suddenly it goes up. So this difference between these two lines denotes the strength of the immunoprecipitated dataset. If we see something like that, then it means that we have quite strong signal in the immunoprecipitated dataset that we analyze. Okay, let's do this. So the tool is called plot fingerprint. Okay, so here we have one kind of cells. All our cells are G1E, as you see. If we could have more cells, then we would do that separately for the different kind of cells. So I select here all the 4 files. Again, sample order does not matter. And here I click on the advanced options just to set the bin size. Let's put some small number, 100. And probably everything else will be fine. Let's see, somewhere there is to ignore the zeros. So here skip zeros, yes, not to include bins with 0 reads. Okay, and now we are ready to go. Run 2. And let's wait. Okay, and now we have the results. So 29 and 28, which are the let's see which ones they are. 29 and 28. Let's see. So what we expect is that these two files should be the immunoprecipitated files. So this file should have the binding sites of the tau1, while these ones are closer to the diagonal line. They should be the control. Now let's see if this is the case. Uh, 28 is this one, and this is okay, but something strange is happening here. "26" is the blue one. Now what we see here is that the enrichment actually, so the immunoprecipitation actually didn't work so well, because okay, this is greater than this one. The green one, which is the immunoprecipitated, is higher than the 28 with 26, higher than 26, which is here, and 29 is higher than 27. So these are the two pairs. But we see actually that this input file does not have so much difference than the immunoprecipitated, which means that maybe the immunoprecipitation didn't go so well as we want it to be. Okay, now again, this is a smaller data set. Maybe things will be different if we could have chosen the whole data set, but in any case, these are the results and we should be a bit concerned. In any case, we will continue with the binding size identification with the MAX2 in the next step. Now, I will stop this video here because all these were quality controls that we saw, and now we will go really to the peak identification to find the binding sites along the genomes.